Hello. All right, welcome back to the last session of today, which is on antibody discovery. And I think it's a great personal pleasure to introduce one of the real champions of the antibody universe to you, Jane Osbourn. And Jane has earned her stripes in the early days of phage display when everything was magic and everything was new and exciting and challenging at Cambridge Antibody Technology, of course, where many of the developments and discoveries were made that we use today as if they've always been around. But I clearly remember that wasn't the case in the 90s while these approaches were being explored. So Jane was at the middle of all that. She went on to MedImmune in senior positions, leading their antibody discovery efforts and development efforts, and now is writing another chapter to her book, which is at Alchemab Therapeutics, where she's going to tell us all about today how to further explore the immune system to come to even better and more differentiated antibodies. Jane.
Great. Well, thanks, Tom, for the introduction, and thanks for the invitation to be here today. And I must say, I've already learnt a huge amount through the presentations over the course of today. And I think one of the themes is the progress that's been made in terms of designing antibodies and some of the amazing stories that we've heard. For us at Alchemab, nature, we think, is one of the best engineers and the best designers. So what we're trying to do is harness nature's answer to dealing with disease by looking for protective autoantibodies. And I'm going to tell you how we're doing that. And Yanni, I think, mentioned, you start out with maybe something that sounds a bit like nonsense and turns interesting. We're early in the journey, but I think we're starting to see some interesting results coming out of the platform that we've been building at Alchemab. So what we're doing is, our vision is to generate novel therapeutics by focusing on protective antibody sequences that are found in people who do unusually well in their disease. So really thinking about wellness rather than illness. So we look for antibodies that are convergent, that are shared in resilient individuals. And I'll tell you more about how we do that. And so this gives you the antibody. It doesn't straight away give you the target. It's target agnostic. So we then have to go and work out, okay, of those shared antibodies, what are they binding to? And we've built some advanced computational black box magic tools as well to help us do this, which I will tell you a bit more about. And we are focused on the BCRs, so the humoral adaptive response, and it's great actually for me to stand up and talk after Aridem's really excellent talk on TCRs, and somebody asked the question, what about the BCRs? This is the answer to that question. So we're looking at the adaptive response, and we're really looking at how we can harness that. And again, I think the sort of ground has been laid by various people today, really explaining that the B-cell repertoire is hugely diverse, made up of boys and girls, I think, and all sorts of different flavors in between. The sum of all B cells expressing distinct B cell receptors necessary to bind diverse antigens to protect us against disease. And that gives you an effective humoral response. And the B cell repertoire has two elements. So you have your primary repertoire, which is antigen independent. And then on antigen challenge, you then build out your secondary repertoire, which is antibody dependent. And there you go through your somatic hypermutations, you build out your plasma cells to deal with the response, and then you build your memory cells as well. So for the primary repertoire, and again, I think it's been quite eloquently explained in previous talks today, this is all about combinatorial diversity. And this audience knows very well that the BCRs are made up of lots of different elements which are combined to the variable gene segments in the heavy chain. You've got over 40 of those, 23 D segments, 6J, all combined with additional diversity in terms of the joining. And then you have a similar approach in the light chain. So you can debate really how big the diversity of the repertoire is. It's certainly at least 10 to the 13, 10 to the 14, potentially significantly higher than that in the primary repertoire. And then you have your antigen response from your secondary repertoire. So you have antigen binding some of your primary repertoire. The B cells activated go into the germinal centers where you have proliferation, hypermutation, and very importantly, class switching. So you're moving from the early IgM response through to an IgG or an IgA response. Selection of those high affinity antibodies which take you to the memory cells and the plasma cells. And there's been some really significant advances over the past decade or so around being able to understand the diversity of B-cell repertoires through high-put sequencing. The first BCR repertoire was sequenced in 2009. That was actually from zebrafish. And since then, there's been a lot of really brilliant work and very technical work building out our ability to sample BCRs. And actually, if you start sampling them properly, they're actually significantly larger than the genome. So it's a lot of next-generation sequencing. And again, Aridam and the TCR talk kind of laid the ground for this, but because you have this amazing diversity, it is actually really extremely rare to find antibodies that are shared between individuals. I've talked about the diversity, the theoretical diversity of around 10 to the 14. At any one time, you've got around 10 to the 9 B cells circulating in your periphery, so you're sort of sampling that diversity all the time. There was a very nice paper back in 2019 from a group at the Scripps where they asked this question, how common are shared BCRs? And they sequenced 10 individuals and found that between 2 individuals, there was some shared public convergent clonotypes, less than 1%. But once you took that to more than 2 people shared, then that number goes down significantly. So only 0.02% shared between all 10 individuals. And again, those shared clones tend to be the short CDRs, where it's more likely that you're seeing that convergence. And also you do see some shared BCRs as the result of vaccinations and common infections. So convergence is rare. That's a key message there. So we're looking to find convergent antibodies. And we're looking to find autoantibodies. And generally people view autoantibodies as the bad guys because people go to the doctor with autoimmune disease. So SLE, MG, various other diseases are caused by pathogenic antibodies, and the treatments for them tend to be, well, ablate the B cells in some way or block the antibody binding. But if there are bad guys, then the immune system is all about balance and yin and yang. There potentially could also be good guys. Just that the good guys are harder to find because you don't go to see a doctor if you have protective antibodies because you're resisting disease. But there are some examples from the literature that are quite nice ones actually. And maybe just focusing on the graph on the right here. This is a survival curve for patients who have naturally occurring anti-HER2 antibodies, and they have a significantly improved survival in breast cancer compared to those patients that don't. So you can find naturally occurring protective antibodies. So if you put all those pieces together, that takes us to the concept that we built with our company Alchemab, where we start by identifying really resilient cohorts of people who are unusually well. So, and I'll explain more about that, but they could be a long-term cancer survivor, a group of cancer survivors, and we do very, very deep sequencing of their BCRs. Deep enough that we can look and see, can we see any antibodies that are convergent, shared in those resilient individuals, and are not found in progressors, and we're able to sort of subtract all the healthy control, natural immunisation, antibodies away from that as well. And we do all of that in silico, and that gives us a relatively small number of antibodies that we then go on and make, and then that we get to really hard biology of trying to understand what do those antibodies bind to, and does this give us insight into a protective effect. So you really are taking an unbiased approach. So this is really turning drug discovery a little bit on its head. I've spent many years working in target-directed drug discovery, and it's been very enjoyable and very successful. But I think, sort of thinking of the high-hanging fruit graphic we saw earlier, we need to start thinking differently about drug discovery. So this is a way of saying, okay, nature, what is your answer to this disease? So defining resilience is an interesting question, and it's ongoing, and we're learning all the time. In cancer, we look for people who are long-term survivors, or are have an unusually good response to treatment. We can look at people who survive infectious disease, people with predisposition to neurodegenerative diseases who don't develop them or progress unusually slowly. And then the ultimate resilient population is the really healthy, long-lived, and we've actually got access to a super centurion cohort who are giving us some very interesting data as well. We couldn't do this without some really open-minded and generous-spirited collaborators who are prepared to take a risk on what is a novel concept. And I did just want to put this up and thank everyone who's had the confidence to share really precious patient samples to enable us to do our deep sequencing. And we've got a range of collaborators in neurodegenerative diseases, oncology, and also in platform and in the long-lived cohorts. So how do we identify the convergent antibodies? So we do this really deep population repertoire analysis, and as I said, we sequence the disease-free population, the resilient patients, ideally between 10 and 100 of them, and the progressors, and then we look for the small area of convergence, which are just seen in those resilient individuals. In terms of what that looks like as a sort of numbers in the funnel, say we have a cohort of around between sort of 10 and 100 individuals, we might generate 10 million sequences. What we then do is cluster those sequences based on, initially on sequence identity or very high homology. We group together antibodies that are highly related. And then we look for the antibodies that are convergent in the resilient individuals, and that sort of takes us down to possibly around about 1,000. And from there, we then go on and really sort of use our antibody engineering knowledge to think, okay, of those 1,000 convergent clusters, which ones of those are highly somatically mutated, so that there's some active selection going on, which ones are class switched, and also going back to thinking about manufacturability and developability. We do have a lot of knowledge, classical drug discovery knowledge around sequence liabilities and sort of, some of the thoughts that Peter presented earlier, so we can actually choose to not take anything that we might have concerns around manufacturing. So we do our repertoire analysis. We also look at serum from these resilient individuals to look for proteomic signals which might help us identify antigens. And we've been building out proteomic tools for target deconvolution. And then we have a proprietary set of deep learning algorithms that can enable us to mine the data set, look for iterative examples of convergent antibodies, and also start thinking about looking for shared paratopes as well. So this was sort of really early in the evolution of Alchemab. We just did some high-throughput sequencing of some different diseases to see, do you see different BCR fingerprints in different diseases? And this isn't unique to anything that we've done. Plenty of other groups illustrate this, but this is some of our own data showing each column is an individual. The kind of heat map is VH germline usage, and you see a very different pattern of usage in a panel of COVID patients compared to influenza, compared to a small cohort of breast cancer patients that we'd seen. So you certainly see different BCR fingerprints in different diseases, and that wouldn't be too much of a surprise to this audience. But what can you do when you start sort of digging in and looking to see if you actually find convergent signals? So I'll go through a few case studies where we've done this. And really the first example is infectious disease, and this is sort of the simplest case, if you like, because you're going into a population. So we managed to deep sequence a series of patients who'd recovered from COVID relatively early in the pandemic, and we published on this in Frontiers in Immunology. But you know when you need to sample those patients. So within 2 to 4 weeks of the infection, we're doing our deep sequencing, and we're finding in the COVID patients actually a reduced clonality. You can see in the scatter plot on the left there. And when we look for convergent antibodies in the recovered patients, we could screen for viral neutralization in this case because it was a fairly obvious functional screen to do. We did indeed find antibodies shared in these individuals that neutralize viral infection of SARS-CoV-2 and actually did a pretty good job of broad neutralization compared to some of the commercially available antibodies that were already in development. So, you know, in terms of an infectious disease proof of concept, I think this is really nice. And it's probably worth pointing out that we screened for viral neutralization. We had other convergent antibodies from this piece of work that weren't necessarily binding the RBD. They could be binding host proteins, and we haven't gone and mined those, but I think there's an interesting avenue there in infectious disease. So infectious disease is one thing. Neurodegeneration is a much more challenging indication. And it was actually great to hear Joe from Denali's talk and Sanders on some of the really creative ideas around how we could potentially address neurodegeneration. We all know that there's a massive need in neurodegenerative diseases. We're still testing some quite well-trodden biological hypothesis, and I think that Sanders made the point very nicely. The animal models are not predictive. So to take a different approach, we feel is really potentially valuable here. And we believe that resilient patients could provide genuine novel insights into how the body fights off disease. So we're sort of physiologically validating these targets. And we know the need. I mean, I think, but I think the point here is that we're initially focusing on Alzheimer's disease, but this is part of a spectrum of dementias and also a broader spectrum of neurodegenerative diseases where we do need to come up with some modifying therapies. So how do you define resilience in Alzheimer's disease? So we've done this in a number of ways, and we've ended up actually combining all of these definitions of resilience in some of the later work that we've done. But you can look at rate of cognitive decline using mini-mental state and other cognition scores. You can look for predisposition to Alzheimer's, looking at APO status or other genetic mutations. And you can also go in and look for biomarkers that will be indicative of Alzheimer's and not seeing it. So high amyloid, low tau is quite an interesting one. And there are some nice literature examples of extraordinarily resilient individuals. This is an example on the right of an individual with a presenilin mutation who had very significant sort of 20-year delay in any cognitive impairment. So we can find these patients. The first study we did, we actually accessed a set of patient samples from a commercial supplier, and we were initially focused on that slow decline based on cognition, which at the end of the day is the sort of ultimate readout for Alzheimer's. So we had a panel of 13 slow progressors, some fast progressors, average progressors, and some age-matched controls. And we went on and did our deep sequencing. If you just look at a high level at the repertoire of these different groups, you can see in pink we've got the Alzheimer's convergent clones. So these are clones that are convergent in the resilient patients compared to the healthy control. And you see changes in V gene usage, which potentially gives you some indications around where you should be looking for your interesting antibodies. And we have a way of identifying convergent clones based on the clonotyping, exclusivity, we look at expression levels, and we have a mutation filter as well. So that funnel in real life for this example, we had over 6 million sequences. We found a couple of hundred convergent clusters shared in the resilient in the resilient patients. And we chose just around 38 of those for production. So all in silico up to that 38, and then you go into biological screening. So we look for antibodies that bind neurons, astrocytes, microglia. We do immune phenotyping and profiling. And we found some really interesting hits already. So antibodies that are binding immune cells and that are binding astrocytes. And just to give you one example of one of the convergent antibodies, this binds to a neuronal target. So our staining antibody is in pink, and this is overlaid with a classic presynaptic marker called vGlut1. And you can see that we're seeing presynaptic staining of this antibody. We don't know what target this is yet, but it's certainly very specific to neuronal cells. So, just to sort of reflect on that, we've gone from over 6 million sequences. We've chosen 38, and one of them gives you this really nice specific staining. So we need to start building out understanding the functionality of these antibodies, and we're doing that at the moment. So that was the sort of starting point with Alzheimer's. We've gone on and managed to access resilient cohorts from frontotemporal dementia patients, Huntington's and Parkinson's, and we've now identified a number of targets. We're just sort of showing 4 here, where we're going into functional assays, and we're hoping to get those into later stage pharmacology over the next year or so. So that was neurodegeneration, and just sort of thinking about it, that neurodegenerative diseases are really chronic, so you're looking at probably a couple of decades worth of immunological response. So the fact that you can find convergent antibodies in that setting, I personally found quite surprising and very exciting, and I think we'll start mining those in more detail. Oncology is kind of between neurodegeneration and infectious disease. You've probably got an immunological response happening over a few months to maybe a couple of years. So we're also really interested in applying the platform in oncology. And for this, we've taken long-term pancreatic cancer survivors. So it's very rare to survive pancreatic cancer more than 5 years. And we've managed to find a group of 12 resilient individuals who are more than 7 years out in terms of survival. They've all had primary resection, but sort of a range of ages. And we know quite a lot about the patient metadata here. And when we, again, went through our process of deep sequencing the BCR repertoires from these patients, we found some really encouraging results here. So this is quite a sort of detailed table, but this is really demonstrating, so on the left you've got the sort of size of the clusters that we were finding, so relatively high expressing clusters, and for some of these clusters, 8 out of 12 of those resilient individuals had this antibody, had this sort of related CDR. So, and you can see that these are actually quite long, some of these are long CDRs as well. So sort of going back to the potential of finding these are identical VHCDR3s in 2 different individuals, that is incredibly rare. So something is going on here in terms of biological selection. And we're also seeing quite a lot of class switching as well. We're working on identifying what these targets are. One of them was one of the antibodies that we found were actually reasonably homologous to Alchemab, which is an anti-VEGF2 antibody. And when we tested our antibody for binding to VEGF2, we did indeed find that it was binding to VEGF2. So in some ways, this is a nice proof of concept that we've gone and found a therapeutically relevant target by going through this route. But we are really sort of excited to learn what other maybe sort of more novel targets we can generate through this, and actually understand what are the natural epitopes that we might be selecting for. So that's sort of case studies, infectious disease, neurodegeneration, oncology. What we've generated by taking this approach, I've just given you a flavour of some of the diseases we've looked at. We've now got the largest BCR sequence database, I think, in the world in terms of association with resilience. So well over 100 million BCR sequences now. And we've got quite a lot of patient metadata associated with that. We're building out annotations, and we're looking at in vitro data to go with this. And what this can now enable us to do is look across diseases. So we can look at those convergent antibodies from the Alzheimer's population and say, are any of those seen in any other neurodegenerative disease? And similarly in oncology, are any of these antibodies seen in a range of different solid tumours? So I think that has the potential to give us really significant insights into that disease analysis. And it also gives us quite an interesting tool in terms of patient stratification. If you go, remembering the heat map that I showed you, can we use this data to identify patients that don't have those protective antibodies and could potentially benefit from them? Because we have all the data for the progressors in here as well. And we've heard, again, around how you can use machine learning to generate really important knowledge from this information. And I think, the sort of descriptions of Aridane's stories of using natural language approaches to question the data, that's exactly what we're doing with the BCR data. So from our database, we can predict, based on natural language approaches and training, which of our BCRs are naive B cells versus memory cells. And there's some interesting little areas where the prediction isn't so good, which we might want to dig into. We can look at numbers of mutations, we can look at germline usage, and we are starting to learn the language of the BCRs in terms of how they associate with disease. So I think this is a really fascinating area to build on. And just a sort of reflection as a sort of end, really. As I said at the beginning, there's been huge amounts of progress on developing antibodies in a target-driven way through fabulous techniques of hybridomas, phage display, transgenics, single-cell sequencing. And I've worked on many of those, and it's amazing the progress that has been made over the past 20 or 30 years in terms of how they've delivered for patients. But they all rely on knowing the target. Even if it's an in silico design, you need to know the target, which means you have a biological hypothesis that you're hanging your hat on. And so what we're trying to do is complement that, really, and think, well, okay, can we flip this round? Can we dig into the natural immune responses and start with the antibody and then try and work back and screen out and find those targets that could iterate back into some more directed evolution as well? But I think this sort of idea of convergence is a very powerful tool to potentially enable that. So I think I've covered all this, really. So protective responses in resilient individuals, but really taking this hypothesis neutral approach in the beginning and trying to sort of build that out. And I'd just like to finish by thanking the great team of colleagues that I have at Alchemab. This is just some of the sort of senior leadership team, but a team of over 60 people in Cambridge, London, and in Boston, who have done a phenomenal job since November 2019 when we were started to bring this idea to a reality and actually generate some really nice data. And I already thanked the collaborators and people that have helped provide us with patient samples. I also want to thank the investors who we're prepared to take a risk on a different idea and actually give us the opportunity to build this platform and to start testing out some of the antibodies that we're finding now in more biological systems. And I will end there. Thanks for your attention. Thanks, Tom.
Thank you very much, Jane, for an inspiring and I think heroic effort to turn around and work without targets, but rather use B-cell very small footprints to find targets and understand disease better and even come up with therapeutics that probably were difficult to address through conventional means, where we indeed understand the targets better and not go from a black box.